Experiment work taking up the vast majority of the work for the astronauts on board the station, and several new experiments studying bacteria were actually launched in return earlier this spring on SpaceX 3. Why don't we now go find out a little bit more about a couple of these from Lori Meggs out at the Payload Operations Integration Center at the Marshall Space Flight Center in Huntsville, Alabama. Lori, I understand results of these experiments could actually help us on future flights to Mars.
Past studies have shown that bacteria becomes more virulent in space. That's why we need further research to understand why as we travel to farther places out like Mars. The experiments we're talking about today are called bric 18. I spoke with David Reed, the lead payload engineer at the Kennedy Space Center, to find out more about these investigations and how the bric canisters work.
Bric 18 consists of two investigations. One investigation is looking at the effects of flying bacteria in microgravity, specifically looking at their how they evolve their resistance to antibiotics. There's a lot of concern right now in the spaceflight community. There's been other research done that shows that some organisms become more virulent, more able to cause disease when they go into space, and part of that equation is understanding how they might become more resistant to antibiotics as well, which could be a real problem on a long-term space flight in some place like Mars. So this investigator has got two kind of common bacteria, and he's applying two different antibiotics to them to see. They each have slightly different genetics in them to see how they respond to that treatment. The second investigator is using a small plant called Arabidopsis. It's in the mustard family. It's a very small plant. It only grows about this big typically before we halt these kind of investigations. And it's a so-called model organism because a lot of work has been done on the plant. Its whole genome is sequenced. But in any event, this particular investigator is looking to understand the effects of spaceflight stress on how that plant expresses its genes. So when a plant feels a stress, like a heat stress or a water stress, it expresses certain genes that make certain proteins in its body, and then we can arrest its development, which in plant terms means we fix them and bring them back home, and then afterwards we can do biochemical analysis to understand how those genes were turned on and off. So 6 of these guys fit inside of a much larger canister that looks like this, and this is called the bric PDFU canister, and you can see there's 6 positions that are each identified with numbers 1 through 6, and that's because the crew member may be asked to chemically preserve individual positions at different points in time. He does that by taking another device that looks and acts a whole lot like a fancy caulking gun. And so the way the fancy caulking gun called an actuator tool works is you pull back the pommel on it, again just like a caulking gun, and then this mates with the top surface of the canister like this. And in the process of a crew member depressing the actuator, then you can see that this piston travels down. In doing so, there's a little bit of engineering magic that goes on inside here. There's a steel rod that gets driven down through some septas. That steel rod is what actually pushes that piston then inside that I already explained is within the individual PDFU, and that forces the liquid on there. Then when the crew member takes this off, at all points in time, this device maintains 3 levels of containment. So containment and keeping the crew safe is, of course, of paramount importance when we fly in space. So, the sort of thing that you could do easily on the ground by simply pouring something from a beaker on top of biology is a little more challenging in space. So there is some sophistication, even though it's something that we call a brick, and it even looks like a brick. You could almost build yourself a fort out of these if you wanted to.
Now, these experiments and several others that came back on SpaceX 3 are now at the Kennedy Space Center for post-flight analysis.